Okay, hello. So today we're carrying on with our lectures on biochemistry and we're moving on to amino acids, and then in the next lecture we're going to talk about the urea cycle and some of the disorders. Shirt check. Today we got Jersey Reds vest, the rugby club from Jersey Channel Islands. So yeah, check them out. Great island. And yeah, let's move on. So at the top we've got some learning objectives that we're going to be going through. But firstly, if we talk about amino acids, where do we get them from? So they can either be exogenous, so get them from food, or endogenous, generated in the body. And these special ones, we've got branched chain amino acids, so leucine, isoleucine, and valine. And they're essential, as we'll look through other essential amino acids and we'll have a chat what they do. But yeah, let's move on. So the essential ones, we've got them here. So the branched chain ones and the phenylalanine. So these are only some of them, obviously there's loads more. So you can have a research if you want. But some of the important features of these, we've got isoleucine as the hemoglobin, glucose and repair, leucine for healing, growth and nitrogen balance, valine for muscle metabolism, nitrogen balance, and then phenylalanine, we've got vitality, alertness, and role of biosynthesis of other amino acids. Then we've got these non-essential ones. So these are basically our DNA bases here. They've got glutamine, acid-alkaline balance, DNA RNA synthesis, ammonia and transport. Then we've got cysteine, infants and certain metabolic diseases. Alanine, nitrogen transfer to the liver, immune system, and tyrosine, hormone precursor, and stimulates metabolism and nervous. So we'll see tyrosine again when we do the thyroid hormone, as that's one of the precursors for that. So if we talk about exogenous protein, so obviously when we eat protein, they're broken down to these polypeptides and then 3 amino acids by peptidases in our stomach, transported into the body by some different transporters. So we've got unisin and antiporters. Yes, as we talked about in the lipoprotein lecture, I believe. So this is kind of the same vibe, where active transport allows for the solute to go against its concentration gradient, where we have this entering of sodium ions and the proteins, and the potassium ions leaves the body to balance out the charge. These proteins can enter via facilitated diffusion, so that means with the use of a certain protein that helps them out to enter the blood and then go wherever they want to go. So if we talk about some of these, firstly, defects that can occur with these amino acids. So we've got this lysine protein intolerance. That's defect in this SLC7A7 gene, and what that happens is the lack of Y plus L amino acid transport 1. It sounds complicated, but it's just a transporter, and a defect in this means certain amino acids can't be transported around the body. Then we've got this heart nub disease, another gene defect of SLC6A19, and that's a lack of protein for system B0 neutral acid transport 1, and that means there's decreased intestinal absorption of tryptophan from the gut. So another amino acid that we need, and with this gene defect we have a decreased absorption of it. So if we talk about now different types of amino acids, so we've got this glucogenic, which have carbon skeletons which are made into pyruvate or Krebs cycle intermediates. Or we've got this ketogenic, which, as you guessed, generate ketone bodies from their carbon skeletons, or acetyl-CoA, which is also very important. So if we talk about this nitrogen balance, so obviously amino acids are a major source of nitrogen, and this nitrogen balance just basically talks about their excretion equals in intake. Some of us can be in positive nitrogen balance, so if we have more nitrogen than we excrete, so such as in growth, when we're growing, and that caused by growth hormone, and we'll talk about that in future lectures. Also pregnancy and tissue repair, because obviously we need to supply our body with that extra nitrogen that we've lost. And also negative that can be malnutrition, starvation, illness, or following surgery. And these amino acids are stored in this free amino acid pool, so they're readily available throughout the body. So if we talk about the liver, that's where most of the action happens again, so that's why you need to keep your liver healthy. And they enter, so amino acids enter the liver, and they're either used for protein synthesis, transamination into amino acids for the urea cycle to get rid of excess nitrogen, because we see that ammonia can have toxic damage to the brain, and we'll talk about a paper in the next lecture. And they can also be broken down further into acetyl-CoA and pyruvate for keto, lipo, and gluconeogenesis. So we talked about that pathway in one of the first lectures, where amino acids can be used for this gluconeogenesis if we're low on glucose. And the amino acid rich blood arrives via the hepatic, so hepatic relating to the liver portal vein. and it's also the only organ capable of eliminating these amino acids by urea synthesis. And amino acid oxidation provides over half the energy of the liver, so yeah, very important. So we talk about catabolism, so the breakdown of, and that basically talks about transamination here. So we've got this AA1, which is amino acid 1, and alpha keto acid, and this basically work in pairs to transfer amino groups between molecules. So we've got this alpha keto acid then 1 and amino acid 2, and this is done by a series of enzymes called transaminases. And a keto acid here is just basically an amino acid without the amino group. So we're basically moving this amino group from one amino acid to another. So we've got these two main ones, ALT and so aspartate aminotransferase and alanine aminotransferase. So the first one's found in cytoplasm and mitochondria of tissues, whereas the second one is specific to hepatocytes. So I always remember ALT, we've got L in this, so it's related to the liver. And these are markers of damage. So if you see raised levels of ALT, that could suggest hepatic damage. Also, for this reaction to occur, we need to have B6, and the active form basically helps to move this amino group, and it becomes an intermediate, so the active B6, and then it allows the amino group to then move to one of these, the other amino acid. So it basically helps them along. And each one of these reactions between amino acids, they have partners, which are important to know. So we've got alanine and pyruvate, aspartate and oxaloacetate, and glutamate with 2-oxaloglutarate or alpha ketoglutarate, the same thing here. So yeah, these amino acids have the keto acid partners here. Then we talk about in detail about these branch-chain amino acids, so valine, leucine, isoleucine, and they're basically mainly used in muscles. So when after a meal we have an increase of these in skeletal muscles, because they possess this brown chain to oxoacid dehydrogenase. It sounds complicated, but it just basically breaks down these brown chain amino acids, and these can be transaminated and oxidized into energy. So what happens is these aminotransferases turn these brown chain amino acids here, BCAA, into the intermediate form. Then this BCKD enzyme decarboxylates them, and we have a formation of acetyl-acetyl-CoA and propanol-CoA, and with this reaction energy is produced. So a quick note on that hormones control all of this. So insulin stimulating this amino acid synthesis in muscles, but inhibits protein to amino acid synthesis. We talk about that insulin is our storing hormone, so it wants to store things. It doesn't want to break down these proteins into amino acids. So if we talk about deamination and ammonia, so how do we then get rid of this ammonia that we've made? And that's again done in the liver. So a lot of reactions end with this glutamate in order for this NH3, sorry, this would be just NH without the plus, can be removed. And we've got glutamate dehydrogenase that can turn glutamate into alpha ketoglutarate and this ammonium ion here. And this can be then put into the urea cycle, which we'll talk about in the next lecture. Then we've got this fixation of ammonia. So glutamate can be made into glutamine through this glutamine synthetase, and this uses ATP to incorporate that NH3 group into glutamine, because we can use this glutamine in other reactions as well. And glutaminase reverses this reaction when it combines with glutamine with water to make glutamate and NH4+. It's basically, we've got this glutamine which can serve other purposes in the body, but then when we have excess, we can use this glutaminase, turn it back into glutamate and NH4+, and then get rid of it through the urea cycle. So the body has a very balance. So we obviously, again relating to this nitrogen balance, need to find a midpoint between these in order so we don't have too much of ammonias that can be toxic. So yeah, that's a quick overview of amino acids. So don't forget to check the description for my links if you want to get free trials. We've got Amazon Prime, music, and other stuff like that. And there's also links to the books that I've used. So yeah, leave a like. Hope you enjoyed. Subscribe for more. Cheers, guys. Cheerio.